Good afternoon. This is Dr. John Bennett, broadcasting from Miami for the Hangout channel of MedGadget.com TV. Today we have the pleasure of having Kimberly Ahmad Schifferly. Is that right, Kimberly?
That's correct. Yep.
Associate Professor of Department of Engineering at UMass, and she's going to talk about cheap paper tests to screen patients for Ebola, yellow fever, and dengue. A very interesting topic. And today we also have a guest panelist, Gene Ostrovsky, the editor of MedGadget itself. Hello, Gene.
Hi, how are you?
Very good. How are you doing today? You're out in San Francisco, right?
Yep. And thank you, Kimberly, for coming.
Great. Thank you both, John and Gene, for having me.
Very good, Kimberly. Okay, why don't you just say a few words about yourself, a little bit about your background, and then talk about the project, and then it's all yours.
Okay, sure, great. My name is Kim Ahmad Schifferly. I'm actually just started as an associate professor of engineering at UMass Boston, and I was previously at MIT in the departments of mechanical engineering and also biological engineering, and my background's in chemistry, so I was really trained as a chemist, but as you can see, this is kind of an engineering type project. And the work really is, I'd really like to point out that this work is a collaboration of many people, so I'm just one person who's on this. We've been working on it with a huge team of people from different backgrounds, from Harvard Medical School and of engineers and virologists and people who study infectious disease, and myself. And so this is something that's only been able to really come to fruition with the effort of a lot of people. So that's something I'd really like to be very clear about. So I guess I'll talk about the work itself.
Yes, please.
And just a quick question, like how much time do you think I should devote to?
Well, 20 minutes, 25 minutes if you want, then we'll have some questions. Is that okay, or 15 minutes is fine?
Yeah, that's fine. That gives me a good sense of what to do. So
okay.
So basically, before I start, this is something we've been working on for 5 years. We're really just interested in making a infectious disease diagnostic that could be used in underdeveloped and countries with poor infrastructure. And so a way to tell if somebody has a disease very quickly on the spot in a way that is can be operated by a non-expert. And it's something that there's a very pressing need for. We definitely saw this with the Ebola outbreak. That's one example of an infectious disease where they really needed a rapid diagnostic, and they really needed it like quickly. And so we are interested in delivering something like this, not just for Ebola, but for many diseases. So that's really our goal. We are also interested in not just infectious disease, but other applications where rapid diagnostics are useful, so that you can think in your everyday life. Sometimes a rapid diagnostic is necessary for things like, we use them for things like pregnancy tests and whatnot. We're also interested in things like food safety and others. But infectious disease is the focus of the work I'll talk about today. So. Okay, so I guess with that, I'll launch into my slides, if that's all right. Yeah, okay. So I'm going to screen share.
Okay, we'll give it a good shot. Very good. You got it the first time. Very good.
Great, excellent. Okay, so pretty much this is something that we wanted to do using. We wanted to do a different take on a rapid diagnostic and make something that was multiplexed, so something that could detect multiple diseases simultaneously. And what we wanted, the punchline of the story is that it was something that we wanted to do in a multicolored fashion, so be able to distinguish between different diseases in using different colors. And we used silver nanoparticles in order to achieve this. So this is something that was press released very recently at the ACS conference in Boston in August. Okay, so we know that infectious disease is a big problem. Dengue is one that we started looking at in particular because the colleagues I worked with we're just interested in the molecular mechanisms of dengue. But pretty much it is a major global threat. It's a mosquito-borne disease, so it's spread by mosquitoes. If they bite you, then you become infected with it. And the really massive problem about it is that it has a huge disease burden, in that 2.5 billion people are at risk of dengue. So the number of infected cases for dengue have been growing. So if you look on the maps from where dengue was in the 70s, you can see it was kind of confined to these sort of small areas in the tropics. But that has grown very rapidly. So if you look at more recent maps from from the CDC, you can see that it's spread all over tropical areas and usually places with poor infrastructure, like the Philippines, India, a lot of places in South America, and so forth. So there's probably about 500,000 people who are hospitalized for it per year, and the fatality rate is about 5%. So there is no specific treatment for dengue, and there is a vaccine in development, but we're still years away from it being deployed to many people. So, and some people view it as a potential biothreat agent. So because it's spread by mosquitoes, it's usually in areas like this is a picture from a New York Times article about dengue. This is in Calcutta. This is an urban area. They have very little infrastructure for running water. So you have these pools of standing water where there's a lot of trash. Okay, these are places where dengue can be very rampant because you have mosquitoes that can breed in this area. Sorry. Yes?
Sorry, I'm not getting the updates for the slides. John, are you getting the updates? Just wanted to check.
Are you trying to move the slides, Kimberly, or not?
No, I'm on the first slide. Is that
okay? You're still on the first slide. Okay, that's good.
Can you guys see that, or is that okay? Okay, I'm moving the slide now. Is that?
Yeah, go at your own speed. That's okay.
Okay. Dengue, a big problem with dengue is that people need a diagnostic because the initial symptom is fever. So pretty much you have a patient who comes in, they have a fever, and you need to be able to tell if they have dengue or if they have the flu or malaria, and it's very difficult to be able to differentiate between those upon initial screening. Okay, typically the way that people have done this is to use PCR and ELISA, but a lot of these places where you have dengue, like in this area, you don't really have access to those resources because they don't have controlled lab environments, they don't have electricity, they don't have running water, they don't have an expert who can run the machine. So that's one problem, infectious disease of interest. Obviously, we're also interested in Ebola. We saw with the outbreak that a rapid diagnostic was really badly needed because and this is a different situation. Now you have human-to-human transition. So you have somebody who presents with a fever, that's the initial symptom, and you can't tell if they have Ebola or if they have malaria, which is non-human-to-human transmission, and you want to be able to tell the difference between those two diseases. Okay, so what the problem with Ebola is that pretty much everybody got quarantined anyway if they had a fever, and so people who didn't have Ebola were quarantined with people who did, and then they had an increased chance of contracting the disease. So quarantine was a big problem, and a rapid diagnostic there would have helped greatly with confining the epidemic. So that's another need for a rapid diagnostic for an infectious disease. So there are many ways to tell if somebody has been infected with a disease. The real state-of-the-art way to do it is PCR and ELISA, as I mentioned before. But we are interested in lateral flow tests because they have the ability to be made very cheaply. They're really easy to use. They require no special reagents, and they can be used by the end user. So you don't need a technician to do this. So this is the same exact technology that's in a pregnancy test, and this is something that is very obvious to use. You open up the pregnancy test, you know exactly how to use it, and the readout is pretty rapid. So it's the same principle here, but I think it's worth going over how the test operates. So what you do is you introduce a sample, and you want to be able to tell if there's a certain biomarker that's indicative of infection in that sample, and so you introduce it to the sample pad, and then basically it just wicks through the sample strip due to capillary action. So if that biomarker is present, then what it will do is it will bind to gold nanoparticles that have on it some species that can bind to the biomarker. So this is a complex, and then this complex will wick through the rest of the sample, and then painted onto the test line is another species that can bind to the other part of the biomarker and accumulate those gold nanoparticles. So this is what we call a sandwich assay, and basically it's because that biomarker is sandwiched between two species that can bind to it, one of which is tethered to the strip, the other of which is tethered to the gold nanoparticle. Now, gold nanoparticles absorb very strongly, so they have a very strong color. So because of that, the gold nanoparticles will accumulate at that test line and then result in a colored line. So basically that gives rise to those colored lines you see in a lateral flow strip. And then to make sure that the flow of the test occurred correctly, there's an antibody painted down on the control line, and then that tells you if the nanoparticles flowed correctly. So basically all you do is you read it, you look to see if there's two colored lines that are present, and that tells you if it's a positive test. If the test line is not there, then you don't have the disease.
Okay. You're still on the first slide, Kimberly.
I'm still on the first slide, actually. Oh. Yeah. Really? That's the one with the picture of the vials?
Oh yeah, I thought that's where you wanted to be. That's okay.
No, I didn't.
Right,
okay.
Let's continue.
Maybe I should. I'm gonna stop and restart it. Is that all right?
Sure.
Yeah. Let's see if this. I know what I'll do. Okay. So, is this? Do you see the picture of the lateral flow test now?
Yeah, lateral flow test, right.
Okay, great. Yeah. So that's basically You know what I'll do? I won't do this. It was the screenshot that was messing everything up, I think. So, okay, so this is the lateral flow test, and you can see that basically this colored test line that shows up is due to the presence of the nanoparticles. Okay. So typically what you do is you just add the sample, no other special reagents, and then it runs through. And this whole thing doesn't have to be refrigerated, so there's no cold chain required to transport them. These can be manufactured in large quantities, and people already have done it for diseases already, but there is none that exists for dengue, and people are trying to develop ones for Ebola, and we saw some examples of it in the literature already. Okay, so now do you see the next slide with the multiplexing by multicolor?
Yes.
Great. Okay, I think the slide advancing is now working. Okay, super. So every test that you see for lateral flow tests are pretty much just one color. They're usually just red. And what we wanted to do was and they usually just test for one thing at a time. So what we wanted to do was to multiplex this test. In other words, have it be able to test for multiple diseases simultaneously. Okay, and this is really important if you want to be able to differentiate between different diseases that somebody may have. So if somebody walks into the clinic and you're not sure if they have Ebola or malaria, or maybe they just have the flu, you want to be able to test for all those from a single sample, okay, in a rapid way, because doing sequential tests make it less convenient. So what we wanted to do was to multiplex it, and multiplexing has been achieved in different ways already. Other people have explored ways to branch that paper so it has multiple branches, so you can have different biomarkers tested in different lanes, and that is something we also are exploring as well. But we wanted to investigate whether or not you can differentiate between different diseases by the color of the line that shows up. So instead of having it be red, why not have it be a different color depending on the disease that you have? So we did a proof of principle experiment to see if we could do this, and the way we did it was to take advantage of the fact that silver nanoparticles, or nanoparticles in general, can be made to have different colors just by changing their physical properties, so what size they are. And so we said, okay, let's try to do this where we can make different test line colors and have it be yellow for yellow fever, red for Ebola, and green for dengue, and see if we can exploit the size-dependent properties of nanoparticles in order to do this. Okay, so that's basically the idea of what we wanted to achieve. So I won't go into the details, but pretty much we synthesized silver nanoparticles that could be made with different colors. It's well known that if you take silver nanoparticles and you change their size, and you can see the TEM image here that they have different sizes, it results in solutions that have different colors. So orange and red and green. And you can see that these are very easily distinguishable by eye. And all we did was just change the synthesis parameters, the concentrations of the reagents, in order to achieve this. Okay. And Those colors are very easily distinguishable by eye, so we had to also test if they were distinguishable when you put them down on paper. And so if you just dry those solutions down onto paper, you can still distinguish between the different colors. And then we also did this in a lateral flow strip. So this is the geometry for a typical lateral flow strip. Here is a schematic of it on the right. So it's a sample pad that you introduce the sample to. Here is the conjugate pad. This is the part that has the nanoparticles conjugated to the antibodies. Dried down in them, and then this part is the nitrocellulose membrane, where there's a control line and a test line, and then an absorbent pad that acts as a wick. Okay, and so these are four different lateral flow strips that we've made, and you can see that this is orange because it has the orange nanoparticles inside it with the antibodies. The red ones are here, the green ones here, and this is a mixture of all four, so it looks brown. So you can see that the colors are easily distinguishable on the paper as well. Okay, this is about 2.5 millimeters wide, just to give you a sense. We need, for this experiment, in order to get this sandwich assay where you can detect you can detect that biomarker of interest, you need to be able to put an antibody on the nanoparticle. And so what we did was we basically just let them kind of stick to the surface of the nanoparticles, or you just incubate the two together, and that they kind of electrostatically absorb on the surface of the particle, and this results in a nanoparticle antibody conjugate. So we take the orange nanoparticles, we conjugate them to the antibodies for yellow fever. The antibodies are specifically for a protein called NS1, and it's known that that's present in the bloodstream when you're infected with yellow fever. We take the red particles, we conjugate it to a different antibody for dengue. There's also another same protein called NS1, but it's a different sequence that is specifically for dengue. And then we do it for Ebola. We take the red particles and we put glycoprotein, viral glycoprotein on that surface, antibodies for that, and that's the other bioconjugate. And pretty much from there, we just verify that those antibodies are on the surface of the particle using techniques such as DLS and zeta potentiometer. Basically, we look for a change in the size and the charge of the species. Okay. So then what we do is we make sure that each of these individual nanoparticle antibody conjugates can detect the antigen individually in human serum. And so we run tests of different concentrations of the biomarker. Here's an example for the yellow fever one. And pretty much, again, we're looking for the presence of this test line shown here. And we can see that as we wick the sample through, if that yellow fever biomarker is present, it results in a colored line on the test line. Okay. We also get a color line on the control line as well. As we decrease the concentration of that biomarker, we can see that that test line fades away, and pretty much what we benchmark as to when we first can detect it, and that's our limited detection. So we do this for yellow fever. We do it for Ebola. You can see that now everything is red because it has the red particles, and we do it again for dengue, and this time it's green because we use green particles. And the limited detection for all these tests is in the order of 150 nanograms per mil. So just to give you a sense of is that high or is that low, for dengue we can compare it to the levels that have been present in the bloodstream of individuals, of infected patients, and it's much, much higher than what's present. So that's typically on the order of 15 to 15 micrograms per mil from the literature, and we can detect much lower than that. So this is good. For Ebola and yellow fever, unfortunately, people don't know those numbers, so we can't benchmark it against patient concentrations. So that's something we're hoping somebody will publish a number on that. Okay. So then what we want to do is to make sure that we can detect these different diseases in a multiplexed manner. And so what we do first is kind of what I call the intermediate step. We spatially separate the lines. So here's our test strip that we are trying to detect. And what we do is we paint down antibodies specific for each disease, but we spatially separate them first. And this is just to make sure that everything works correctly. So on this position, we paint down antibodies for yellow fever. On this one, antibodies for Ebola. On this one, dengue, and then up here, a control one. And basically, we run samples through and make sure that we get a hit at the correct position and that it's also the correct color. So, for example, if we run yellow fever sample through here, we should get a hit on this position only and not here, and that hit, the band should be orange. If we do it for Ebola, we should get a hit here, but not here, and it should be red. And then dengue, it should be only here, and it should be green. And if we have all three present, we should get hit light. We should have it light up at all three positions, and they should be all different colors. Okay, so these are the results of those experiments. We can see that if we run yellow fever, we get a hit here, but not here and here, and it's an orange band. For Ebola, we get a red band here, but nothing else here and here. Same for dengue, we get a green band here, but not here and here. And then finally, if all three are present, we get hits at all positions, and they're all the correct colors. Most importantly, if we run a mock sample that has human serum that has no biomarker for any of those diseases, we get no no positions lighting up, which indicates that there's no non-specific absorption. So this shows us that we're able to be able to run a multiplex test for multiple diseases at once, and we can differentiate between them based on their position and color. Okay, so then what we try to do is to make it harder and try to actually co-localize all those antibodies for the test right here onto one pad. So basically, all the antibodies for Ebola and dengue and yellow fever are all in this position. And so basically, if we should only get a hit at that at that test line only. And depending on what is run through, if the biomarker for yellow fever is present, it should be yellow. If it's Ebola, it should be red. If it's dengue, it should be green. And if it's all three, it should be brown. Okay, and again, for mock, we should have nothing there. So these are the results that I'm showing you right here. If the yellow fever is present, we get an orange band. If it's Ebola, it's red. If it's green, it's dengue. And if it's all three, it's brown. So this indicates that the test worked, and there's no signal for the mock. Now, if you look at this, so my eyes are old, and to me, these colors all look kind of similar, and so you might say, how can you distinguish between, this doesn't really look very orange, and this maybe kind of looks brownish, but no, I'm not really sure. So what we did is we actually quantified this from using an image analysis. So basically, we detected the RGB channels of each of these. And so I won't go into too much detail, but pretty much we do line scans of these, and we quantify the RGB values of each of those test lines. And we can see here that we did this, that we can distinguish it. So pretty much this is the RGB values for each of those tests. And if we run, for example, yellow fever through the sample, it has a certain RGB value, which plots here onto this three-dimensional plot. And the uncertainty is, it's an ellipse. So the uncertainty in R and G and B are represented by the size of that ellipse. And you can see that for each of the different disease biomarkers, you get different positions on this RGB plot, but they don't overlap. So this indicates that the camera on your mobile phone is able to distinguish between these colors if you break it down into the RGB channels. Okay, and it's also distinguishable from both the mock and also the mixture of all three. So this shows us that we can quantifiably distinguish between different diseases based on the color of the line. And so that's basically what we set out to do. So I'll just to summarize, pretty much what we're able to do is to use the size and shape dependence of nanoparticles in order to enable differentiation between different diseases, yellow fever, dengue, and Ebola. Okay, this can help us increase the miniaturization of these tests, so we can shrink down these test lines so we can use less and less sample as we introduce into the device. And then it also shows us that there's not non-specific adsorption in the multiplexed line test. So we're also looking at other particles. So I showed you work on gold and silver nanoparticles, but we're also interested in other nanoparticles that have different colors, like gold nanostars. And there's a lot of work that we're putting into this to try to make this device more efficient. So working on the hardware for this, how do you design the housing, also the fluidics, so all the things about the paper and how the fluid wicks through. There's a lot of work on surface chemistry, as you can imagine, so there's tons of surfaces where molecules can non-specifically absorb, so we're working on those and improving the stability of it as well. We're always looking to the next infectious disease, so other outbreaks that may be occurring, and other diagnostic applications like food safety and so forth. So pretty much that summarizes my talk right there.
Okay, excellent, Kimberly. Well illustrated, exciting technology. And let me just, before we start here, let me just show the viewers where they can ask questions. And Jean put up the tweet board by the side of the screen. If you have a question, here's the tweet board off the MedGadget main page. Just click on Compose Tweet. It'll ask permission for TWUBS to look at your account, but then you can tweet in real time. So, Jean, do you have any questions to start off?
Yes, Kimberly, I wanted to know, is the device reusable or it's a single use?
No, we designed it for single use specifically, yes.
Yeah. Okay, and When do you expect it to be commercialized, to actually do the tests in the field?
Yeah, so we're exploring those options right now. We think it could be on the order of a year or so, or that's what we're hoping. I tend to be conservative in these estimates because there's always a lot of unforeseen things that crop up, and so we're thinking about a year to 2 years.
You know, I also wanted to ask you about like handling and transportation. Are there any limitations in that respect in terms of sending it somewhere in a container that's on a ship for months?
Right, yes, that's a really great question. So, for example, if these are deployed all over the world, they have to go through some sort of transport chain. They have to be stored for long periods of time. And these are issues that can really compromise the performance of the device. So we have started to look at stability tests. So how long can you keep it out in certain conditions, and will it still work? So as with any lateral flow device, they are terrible in humid conditions. So if it's very humid, like in tropical areas where a lot of these diseases are, the test actually starts to run itself, and then it's no good. So a lot of this actually can be solved with proper packaging. So we have done environmental testing with silver foil packets. We basically put the thing in a kind of a food packaging device, and we basically put a silver foil packet around it. And in that case, they are stable at 37 degrees in high humidity for, I think we've tested it out for about 6 weeks so far. So that's they look pretty good.
Kimberly, is the strip immersed in blood, or is a droplet of blood put somewhere on the strip, or how does that work?
That's right. So pretty much you introduce, there's a well, and you introduce a drop of blood or serum, and that is what works through.
Oh, okay. Yeah. Because I guess one of the advantages that the article says about it is that it doesn't need water or electricity, and it can be done in the field by people that really aren't doctors or healthcare people. But how are they going to draw the blood? I mean, that's the question. Like in the field, not everyone knows how to draw blood.
That's true. That's true. That's absolutely true. And so, yeah, that is a huge limitation. Usually pinpricks or finger sticks are very easy to do at clinics.
Sufficient? Yeah. That's sufficient? That amount of blood is sufficient?
That amount of blood is sufficient. You can just basically, what you do is you squeeze out, you prick your finger, and then you squeeze out as much blood as you possibly can. Okay. Obviously, if it's possible to use saliva, that is a much more convenient bodily fluid. But unfortunately, for dengue, the levels of that biomarker are not known in saliva, though some people have started to look at that. As for Ebola, people, we would love if someone could quantify how much, glycoprotein is present in saliva. There's just patient data on that is not available. But if we had that information, that's something we would, obviously design this for.
Okay.
Yeah.
What about other diseases? How difficult is it to transfer this technology to some other disease?
Yeah, that's a great question. And so, in principle, it's very easy as long as you have the antibodies. So if there is another disease where people know that they have antibodies already for a certain biomarker, and you know that biomarker has a reasonable concentration, then this is very easy. The tricky part is not all antibodies work in lateral flow. So antibodies that may work for a disease, they may say, yes, this binds to this biomarker for some disease like malaria or something, but and that may work in a FACS system, flow cytometry. But in our hands, we find that just because something binds in FACS or in solution doesn't mean it's going to work in a lateral flow test. So we pretty much have to throw away a lot of antibodies that don't work in lateral flow.
In Africa, malaria is such a big killer. Will it work with malaria, you think, or something like that, or no?
Yeah, so there actually are. I think there is a rapid diagnostic for malaria already.
Okay.
And I think it does the HRP. There's a protein that an antibody for that protein that's already been developed. So I think that might be already out there. Yeah.
That's covered. Yeah. How about HIV?
Yeah, there are HIV tests that are out there, and HIV sensitivity is actually pretty. People have you detect very, very small amounts of HIV. And I think there are tests that already do that, rapid tests.
Oh,
Yeah.
okay.
Commercial ones available as well. Yeah.
So you're aiming for dengue, yellow fever, and Ebola right now.
Yeah, dengue is the real big one. I think that's something that was our main thing that we started working on, and then Ebola was something that came up, and we had the antibodies. We purchased those in order to go after that. But
yeah, so there's
dengue
nothing.
is the big one.
Yeah, there's nothing out there now for dengue, so that's why you're concentrating on that.
Yeah, there are some but we find that they're not widely available, and they don't have serotype specificity, and they have a lot of crossover with yellow fever. So I think there is a dengue kit from Alere that's currently commercially available, but it's the only one out there right now. Yeah.
I guess there wouldn't be a market for this in the U.S., correct? Because you have to go through all kinds of testing first, FDA.
That's right. Yeah. So with the FDA, it has to be FDA approved. We actually are working with the FDA. We collaborate with them, and so we are looking at the prospect of trying to get it FDA approved as well. But it's a big hurdle. That is very true.
Yes. Gene, any more questions?
Yeah, one more. It seems like the number of diseases you can test for on one device is limited by how many colors you can produce, right?
That's actually not only, but it's close. You can actually, if you branch it out, or if you spatially separate lines, then you can multiply that by even more.
Okay. I was wondering how many colors can you produce, or is it unlimited?
Right. So in terms of the nanoparticles that we can produce them, I think on the order of 10, 15 different colors. So we can make nanoparticles that have different colors that are distinguishable, and so that's definitely possible. Sometimes they start to get kind of close. So like red and orange and green are very distinguishable by eye. It's really easy to tell the difference, and we just wanted to do something that we knew we could tell the difference. But sometimes, like you can have like green and blue-green, and they're really close, and that's.
What if you have like a yellow and a blue, right? A disease that does yellow create blue, and when they're together, does it create?
It looks green. Yeah, that would be bad. Yeah, that would be bad. Yeah.
But there are ways to detect shades of color. You mentioned that. You said you can't distinguish colors well, but there's some kind of way you can distinguish them.
Your phone camera actually is pretty good at distinguishing color. So if you decompose things into RGB values, something that you can't tell by eye, your phone camera can.
Well, I know Photoshop does that too.
That's right. Yeah, it's the same principle. Yeah, it's the same exact thing. Yeah. So we haven't pushed how many channels we can achieve with the multiplexing by color, but that's something we definitely plan on doing. We have made nanoparticles that are pretty close in color, in solution or dried on paper, and then it would be interesting to see how much the phone camera can distinguish between them as well. Yeah.
Very good. Okay, very good. Any more questions, Gene?
No, thank you very much, Kimberly.
Great, thank you, Gene.
Yeah, Kimberly, that was excellent. Thanks for taking the time. And I'm glad
great.
I was getting nervous you wouldn't come, but thank you very much for coming, and I'll send you a copy of this, and we look forward to following up with you to see what else you guys have in store for us.
Okay, great. Thank you so much, John. I really appreciate it.
Thank you. Good day.
Right. Take care now. Bye.